Biotechnology is a branch of biology which deals with the techniques of using live organisms, enzymes, or biological processes to produce products and provide services for human welfare. Making curds or bread, which involves microorganisms, can be considered as the oldest form of biotechnology. We also find application of fermentation in the production of wine and other alcoholic beverages. Most of the biotechnological processes gradually became more sophisticated. They are being used to harvest various valuable materials like vitamins and antibiotics produced by microbes. Today, biotechnology is undergoing resurgence in the wide range of applications. Do you know what all biotechnology involves? Well, DNA manipulations, that is recombinant DNA technology, tissue culture, protoplast fusion, cell catalysis, immobilized enzymes, protein engineering, etc. Many techniques are included under biotechnology. For example, in vitro fertilization leading to a test tube baby. I'm sure you've heard of that. Synthesizing a gene, developing a DNA vaccine, or correcting a defective gene, etc. Advancements in plant tissue culture has enabled plant breeders to create new crops which grow more rapidly, require less fertilizer, and thrive in poor soils. Many techniques included under biotechnology have wide applications in the field of agriculture, medicine, chemical industry, pharmaceutical industry, and environment. The new definition of modern biotechnology given by EFB, that is, the European Federation of Biotechnology, is: biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Recombinant DNA technology. During sexual reproduction, variations are created. Some of the variations are useful. Such variants are not produced during asexual reproduction. Genetic engineering involves a manipulation of the genetic material towards a desired end and in a predetermined way. To engineer means to design, construct, and manipulate according to a set plan. This technology bypasses the restriction in the gene transfer mechanisms between unrelated organisms. It differs from genetic recombination in that it does not occur through natural processes but is engineered. The recombinant DNA technique was first proposed by Peter Loban, who was a graduate student with Adele Kayser at the Department of Biochemistry at the Stanford University. The present-day rDNA technology actually flourished after Stanley Cohen and Robert Boyer in 1972 could successfully link a gene coding for antibiotic resistance with a native plasmid of Salmonella typhimurium with a vector plasmid and then cloning it in E. coli. Recombinant DNA or rDNA technology is the technique of manipulating the genome of a cell or organism so as to change the phenotype desirably. Basic steps of recombinant DNA technology. First, the cell or organism from which the required gene is taken is called donor. Second, fragmenting this DNA using molecular scissors or enzymes. Third, screening the fragments for a desired gene. Fourth, inserting the fragments with desired gene into a cloning vector like a plasmid, cosmid, or phage DNA so as to develop a recombinant DNA or chimeric DNA. Fifth, introducing the recombinant vector into a component host cell. Sixth, culturing these cells to obtain multiple copies or clones of desired fragment of DNA. And seventh, using these copies to transform suitable host cells so as to express the desired gene. Tools used. Enzymes. Different enzymes used are restriction endonucleases, DNA ligase, reverse transcriptase, DNA polymerase, alkaline phosphatases, etc. The restriction endonucleases are used to cut DNA at specific points. They are called biological or molecular or chemical scissors, knives, or scalpels. Vectors are the DNA molecules used to transfer genetic material into another cell. Now we know that the mosquito acts as an insect vector to transfer the malarial parasite into human body, right? It may be a plasmid or bacteriophage DNA or artificial DNA or cosmid. These are low molecular weight DNA molecules capable of multiplying independent of the genomic DNA. They can be employed to make copies of desired DNA fragments. Tools used. Enzymes. The cloning vectors must be able to replicate in the host cell. Thus, it must have a replication origin. A replication origin, do you know what that is? Well, it is a sequence element which is recognized by the host cell's replication machinery, that is, host's DNA enzymes. And hence, the cloning vector can be amplified in the host cell. A segment of foreign DNA which has been inserted into the vector's DNA is therefore amplified along with vector. Large amounts of the desired foreign DNA are therefore produced by gene cloning. Vectors need to have restriction endonuclease, recognition sites, and some marker gene which will express in the host cell. In fact, efficiency of transformation is very low and only a few cells in the population take up and retain the exogenous DNA. marker in vector helps in identification of transformed bacterial colony. Tools used: enzymes. Insertion of a vector into the target cell is usually called transformation for bacterial cells, transfection for eukaryotic cells, or the insertion of a viral vector is often called transduction. Now, some of the common vectors for bacteria are plasmids, cosmids, and lambda-phage. Baculovirus is useful in insects. T plasmid is for plants, and YAC, that is, yeast artificial chromosome, is for yeast cells. Process: DNA taken from both the sources is fragmented by restriction endonuclease. The restriction enzyme cuts both molecules at the specific site. The ends of the cut have an overhanging piece of single-stranded DNA called sticky ends. These sticky ends are able to base pair with any DNA fragment that contains the complementary sticky end. Enzyme DNA ligase is used to covalently link the two strands into a molecule of recombinant DNA. This recombinant DNA needs to be replicated many times, that is, cloned. Now, cloning can be done in vitro via the polymerase chain reaction, or PCR, or in vivo, that is, inside the cell, using unicellular prokaryotes, example E. coli, unicellular eukaryotes, example yeast, or mammalian tissue culture cells. Therapeutic products. Some examples of the therapeutic products made by recombinant DNA techniques include: first, blood proteins, erythropoietin, factors 7th, 8th, 9th, tissue plasminogen activator, urokinase. Second, human hormones, epidermal growth factor, follicle stimulating hormone, insulin, nerve growth factor, relaxin, somatotropin. Third, immune modulator, alpha interferon, beta interferon, colony stimulating factor, lysozyme, tumor necrosis factor. Fourth, vaccines, cytomegalovirus, hepatitis B, measles, rabies. Transposons. Transposons are sequences of DNA that can move or transpose themselves to new positions within the genome of a single cell. They are also known as jumping genes. Transposition can create phenotypically significant mutations and alter the cell's genome size. The first transposons were discovered in maize, that is Z maize, by Barbara McClintock in 1948 for which she was awarded a Nobel Prize in 1983. Now, about 50% of the total genome of maize consists of transposons. In bacteria, transposons can jump from chromosomal DNA to plasmid DNA and back, allowing for the transfer and permanent addition of genes such as those encoding for antibiotic resistance. Multi-antibiotic resistant bacterial strains can be generated in this way. In primates, including humans, some sequences of interspersed repetitive DNA are present. This common form of transposon in humans is called allele sequence. It is approximately 300 bases long. Types of transposons. On the basis of their mechanisms, they are classified into two types. Retrotransposons. Now they copy in two stages. First from DNA to RNA by transcription, and then from RNA back to DNA by reverse transcription. The DNA copy is then inserted in the new position. So it is copy and paste. DNA transposons. They do not involve RNA intermediate. The enzyme transposase makes a staggered cut at the target site, producing sticky ends, cuts out the transposon, and ligates in new position. That is, cut and paste. Plasmids. Plasmids are the small, extrachromosomal, double-stranded, circular forms of DNA that replicate autonomously. Most of the plasmids are not required for the survival of the organism in which they reside. Now, they are naturally found in bacteria, yeast, and occasionally in plant and animal cells. The term plasmid was first introduced by the American molecular biologist Joshua Lederberg in 1952. Plasmid size varies from 1 to over 1,000 kilobase pairs, that is kbp. Plasmids are considered as transferable genetic elements or replicons capable of autonomous replication within a suitable host. Plasmids in bacteria carry genes related to metabolic activity and allowing the carrier bacterium to survive and reproduce under unfavorable conditions. Plasmids cannot accommodate long DNA fragments, therefore, cosmids are constructed. Now, what are these cosmids? Cosmids are plasmids having fragments of lambda phage vectors. It is a common practice to designate plasmid by a lowercase capital P or small p followed by the first letters of researcher, researchers' names, and the numerical number given by the workers. Thus, PBR 322 is a plasmid discovered by Bolivar and Rodriguez, who designated it as 322. Now, some plasmids are given names of the places where they have been discovered. Example, PUC or plasmid UC is a plasmid from University of California. Bacteriophage. In the Greek language, phagein means to eat. Thus, a bacteriophage is a virus that infects bacteria. commonly, its short form phage is used. Typically, biophages consist of an outer protein capsid enclosing the genetic material. The genetic material can be ssRNA, dsRNA, or ssDNA or dsDNA in either circular or linear arrangement. Here, SS or DS prefix denotes single strand or double strand. Bacteriophages are much smaller than the bacteria they destroy. Bacteriophage. The cloning of single genes is usually best carried out using plasmids. However, for cloning of larger pieces of DNA, that is, during gene library construction, these plasmids are not suitable as larger inserts increase the plasmid size, making the transformation inefficient. Larger DNA molecules can be injected in host bacterial cell by bacteriophages. Commonly used bacteriophages as cloning vectors are M13 and lambda phage, which infects E. coli. The portion of phage DNA required for packing it in a capsid is retained and virulent portion is deleted so that desired DNA fragment can be loaded. Phage lambda as vector. DNA of lambda phage is 48.5 kb in length. It is with COS sites of 12 BP at the ends. The cohesive COS ends allow the DNA to be circularized in the host cell. For the cloning of large DNA fragments, up to about 20 kbp DNA of phage is removed and replaced by the DNA with desired gene. The recombinant DNA is then packaged within viral particles in vitro, and these are allowed to infect bacterial cells which have been cultured on agar. Once inside the bacterial cells, the recombinant viral DNA starts replicating. All the genes needed for multiplication of the virus and normal lytic cycle are still present in the DNA. Growth takes place by cycles of cell lysis, and the released viruses infect the surrounding bacterial cells. It gives rise to plaques of lysed bacterial cells on a background or lawn of bacterial cells. Cloned DNA can be recovered from the viruses in these plagues. Replication of bacteriophages, that is lytic cycle, inside the specific host or bacterial cell takes place in the following steps. First, attachment. Bacteriophages attach to specific receptors on the surface of bacteria. As phages do not move independently, they rely on random encounters with the right receptors. Second, penetration. Now, after the contact, the tail fibers bring the base plate closer to the surface of the cell. Once attached completely, the tail contracts, injecting genetic material or DNA through the bacterial membrane. Capsid, that is the protein coat, remains outside and is called ghost. Third, synthesis of proteins and nucleic acids. The host's normal synthesis of proteins and nucleic acids is disrupted, and it is forced to manufacture viral DNA and proteins instead. These products are the parts of new virions within the cell or proteins involved in cell lysis. 4. Virion assembly. The base plates are assembled with the tails first. The heads, capsids, are constructed separately and then they are joined with the tails. The DNA is packed efficiently within the heads. The whole process takes about 15 minutes. 5th, release of virions. Phages are released via lysis of cells. It is achieved by an enzyme called endolysin, which breaks down the cell wall. Released virions are capable of infecting a new bacterium. Restriction fragments. A restriction fragment is a DNA fragment resulting from the cutting of a DNA strand by the restriction enzyme, that is, restriction endonucleases or RENs, a process called restriction. Stewart Lin and Werner Aber isolated two enzymes in 1963 which restricted the growth of bacteriophage in bacterium E. coli. One of these enzymes cut DNA and was named restriction endonuclease. Restriction enzymes belong to a larger class of enzymes called nucleases. Now, the nucleases are of two types. First, exonucleases. They remove nucleotides from the ends of the DNA. And second, endonucleases. They make cuts at specific positions within the DNA. Restriction endonucleases serve as tools for cutting DNA molecules at predetermined sites, which is the basic requirement for gene cloning or recombinant DNA technology. Types of restriction endonucleases. 3 main types, type 1st, type 2nd, and type 3rd are known. They have slightly different modes of action. Type 2nd restriction enzymes are used in rDNA technology because they can be used in vitro to identify and cleave within specific DNA sequences, usually having 4 to 8 nucleotides. More than 350 different type 2nd endonucleases with 100 different recognition sequences are known. Nomenclature. Restriction endonucleases are named by a standard procedure with particular reference to the bacteria from which they are isolated. The first letter, which is in italics, of the enzymes indicates the genus name, followed by the first 2 letters, also in italics, of the species. Then comes the strain of the organism, and finally a Roman numeral indicating the order of discovery. Let's understand this better with a couple of examples given here. Eco RI is from Escherichia coli or E. coli strain. RY13 R and first endonuclease first to be discovered. Hind third is from Haemophilus H influenzae, iron strain, RD, D, and the third nuclease, third, to be discovered. Recognition sequences. Recognition sequence or restriction site is the site where the DNA is cut by a restriction endonuclease. Restriction endonucleases specifically recognize DNA with a particular sequence of 4 to 8 nucleotides and cleave. Each restriction enzyme is highly specific, recognizing a particular short DNA sequence or restriction site and cutting both DNA strands at the same time at specific points within that site. Most restriction sites are palindromes. Now, in a palindrome, base sequence of second half in DNA strand represents the mirror image of the base sequence of first half. Palindromes are groups of letters that form the same word when read both forward and backward. Say, for example, Malayalam. While the same word is read backward and forward in a palindrome word, palindrome in DNA is a sequence of base pairs that reads the same on the two strands where orientation of reading is kept the same. Cleavage patterns. In recombinant DNA technology, specific restriction endonucleases, particularly type II, recognize a specific base pair in DNA called a restriction site and cleave the DNA within the sequence by hydrolyzing the phosphodiester bonds, retaining symmetry, and isolate a particular gene so that the double-stranded restriction fragments have single-stranded ends. These short extensions, called sticky ends, can form hydrogen-bonded base pairs with complementary sticky ends on any other DNA cut with the same enzyme, such as a bacterial plasmid. They require magnesium ions for cleavage. Cleavage patterns. Many cuts are made by one restriction enzyme because of the chances of repetition of these sequences in a long DNA molecule, yielding a set of restriction fragments. A particular DNA molecule will always yield the same set of restriction fragments when exposed to the same restriction enzyme. Restriction fragments can be analyzed using techniques such as gel electrophoresis or used in recombinant DNA technology. In agar-Rose gel electrophoresis, the restriction fragments yield a band pattern characteristic of the original DNA molecule and restriction enzyme used. For example, the relatively small DNA molecules of viruses and plasmids can be identified simply by their restriction fragment patterns. Preparing and cloning a DNA library. In molecular biology, a library is a collection of DNA fragments, specifically genes, from a particular species that is stored and propagated in a population of microorganisms through the process of molecular cloning. Genomic library. It is a collection of all clones of DNA fragments that represent the complete genome of an organism. For construction of genomic library of an organism, its entire genome or DNA is isolated and cut into fragments using suitable restriction endonuclease. These fragments are then inserted into cloning vectors. The recombinant vectors are then transferred into suitable organisms such as bacteria or yeast. One fragment in each host cell, and then these transformed organisms are cultured to produce their clones and are stored.
When genomic DNA is fragmented, it is not known which fragment has the desired gene.
Therefore, all the fragments are cloned and copies of each are stored separately. Screening for the desired gene can be done through complementation or using probes. cDNA library. cDNA is complementary DNA. It is produced using mRNA by the process called reverse transcription. This process is called Teminism, named after its discoverer Temin and Baltimore. Mostly for eukaryotic organisms, cDNA library is constructed. In eukaryotes, there are coding and non-coding sequences in the genes.
The mRNA carrying only the transcribed coding sequences translated into protein.
When mRNA of different cells, tissues and organisms at different time and phases in the life cycle of an organism are isolated, then using reverse transcriptase enzyme, it is converted into cDNA. A library having cDNA for each and every type of structural or functional protein can be constructed by inserting it into a suitable vector and then cloning it in a proper host such as E. coli. Production of human proteins such as interferons, insulin, and blood clotting factor VIII can be done using bacterial cultures having cDNA. Gene amplification, PCR. Gene amplification is obtaining multiple copies of a known DNA sequence that contains a gene. Now, gene amplification can be done artificially by using polymerase chain reaction or PCR technique. Thus, PCR is in vitro technique of gene amplification. It is a scientific technique in molecular biology used to generate billions of copies of a particular DNA sequence in a short time. It was first developed in 1983 by Kary Mullis. In 1993, Mullis was awarded the Nobel Prize in Chemistry for this work. Basic requirements for PCR technique. First, a DNA segment 100 to 35,000 BP in length to be amplified. Second, primers, forward and reverse, which are synthetic oligonucleotides of 17 to 30 nucleotides. They are complementary to the sequences present on the desired DNA segment. Third, 4 types of deoxyribonucleotides, DATP, DCTP, DGTP, DTTP. They are collectively called DNTPs. Fourth, a thermostable DNA polymerase that can withstand up to 94 degrees Celsius. Usually, Taq polymerase isolated from bacterium Thermus aquaticus is used. The vast majority of PCR methods use Thermal cycling, that is, alternately heating and cooling the PCR sample.
Steps of PCR technique. Heat denaturation. This step involves heating of DNA at about 91 degrees Celsius. The heating breaks the hydrogen bonds to make ssDNA. The DNA molecule with more GC pairs need higher temperature. Annealing. It is pairing of primers to the ssDNA segment. The primer have to be designed as per the requirement. This step requires temperature at about 55 degrees Celsius. Third, polymerization. The temperature is raised to 72 degrees Celsius. The Taq polymerase A is DNTP behind the primer on the ssDNA. These three steps constitute one cycle of the reaction. The process is carried out for about 28 to 30 cycles, beyond which its reliability decreases. Each cycle of PCR takes about 3 to 5 minutes. As PCR progresses, the DNA generated is itself used as a template for replication, thus setting a chain of reaction in which the DNA template is exponentially amplified. PCR is carried out on an automated machine. PCR technique is used in medical and biological research labs for a variety of applications. It is used in DNA cloning, gene amplification, DNA-based phylogeny or functional analysis of genes, diagnosis of hereditary diseases, DNA fingerprinting, which is used in forensic sciences and paternity testing, the detection and diagnosis of infectious diseases, and in the diagnosis of cancer. Application of biotechnology in agriculture, BT crops. Biotechnology has also revolutionized research activities in the area of agriculture. To increase food production, there are three options: use of chemical fertilizers and pesticides, use of biopesticides and biofertilizers, organic farming, and use of genetically engineered crop plants, that is, transgenic plants or BT crops. Transgenic plants carrying desirable traits like disease resistance, insect resistance, and herbicide resistance are produced. Plants with better photosynthesis efficiency, nitrogen-fixing ability, improved storage proteins, and higher vitamin content can be produced. Most of the current commercial applications of modern biotechnology in agriculture aim at reducing the dependence of farmers on agrochemicals. Therefore, insect-resistant crops or BT crops are produced. Bacillus thuringiensis or BT. Bacillus thuringiensis or BT is a soil bacterium that produces a protein with insecticidal qualities. Traditionally, an insecticidal spray produced from these bacteria is used. BT toxin proteins occur as inactive protoxins. When an insect ingests it, due to the alkaline pH of the gut, it gets converted into the active form. The activated toxin causes swelling in the gut and death of the insect. There are several Bt toxins and each one is specific to certain target insect. Crop plants have now been engineered using rDNA technology to contain and express the gene for Bt toxin. The gene is called Cry gene. It produces inactive protoxins. When an insect ingests the transgenic crop, it dies. Bt cotton is now commercially available in India to control the disease affecting the cotton bolls. Bt toxin gene has been cloned and introduced in many plants to provide resistance to insects without the need of insecticides. Other examples are Bt corn, rice, tomato, potato, and soybean. Application of biotechnology in agriculture. Bt crops. Agrobacterium tumefaciens is a soil bacterium which causes crown gall tumors in dicotyledonous plants. A gall-producing gene, tDNA, occurs in a large plasmid called tumor-inducing plasmid or Ti plasmid. Due to the ability of the bacterium to insert Ti plasmids into nuclear genome of the infected plant, gene transfer in higher plants through Ti plasmids can be achieved. Tumor-producing gene becomes the maker gene. Therefore, Agrobacterium tumefaciens is most widely used for gene transfer. Cry gene from Bacillus thuringiensis or NIF gene, N2 fixing gene from Rhizobium is cloned inside Agrobacterium tumefaciens and then transferred into other plants. Many Bt crops, transgenic plants or genetically modified plants, that is GM plants, are produced using Agrobacterium tumefaciens. For example, Flavor Saver tomato is developed by introducing an antisense gene that retards ripening. It produces less cell wall degrading enzyme polygalactoranase. Therefore, these tomatoes have longer shelf life. Flavor is saved, which is an additional advantage. Golden rice is genetically engineered rice with greater provitamin A, that is beta-carotene content. Biosafety issues. The commercialization of transgenic crops has sparked off intensive debates worldwide regarding biosafety, ethical issues, and the impact of this powerful technology on agriculture, human health, and environment. Genetic modification of organisms can have unpredictable effects when such organisms are introduced into the ecosystem. For example, cross-pollination between GM plants and wild varieties will lead to contamination of gene pools of wild varieties. Consumption of GM food may develop allergies. You never know, it hasn't been tried for long, right? GM microbes made in the laboratory may be hazardous. Therefore, manipulations of living organisms needs regulation. The Indian government has set up Genetic Engineering Approval Committee or GEAC, which will take decisions regarding the validity of GM research and safety of introducing GM product for public services. Biopiracy. There are issues regarding biopiracy also. Developing and underdeveloped countries are rich in biodiversity, that is bioresource, and traditional knowledge, while the developed nations are rich in finance technology. This leads to biopiracy. Developed countries, since the past 2 decades, have been enjoying immense profits by patenting the knowledge and bioresources of underdeveloped countries. Biopatent. Well, a biopatent is a patent granted by the government to the inventor for biological entities, processes, and products. Patent gives the owner exclusive rights to the use of the resource, process, or to market the product and earn profits. Biopiracy is biopatenting of the bioresource of another concern without its proper permission or unlawful exploitation and use of bioresource without giving compensation. Basmati rice, known for its unique aroma and flavor, has been grown in India for centuries. There are, in fact, about 27 documented varieties of basmati grown in India. Now, what happened is that a Texas-based company got patent trademark office. This allowed the company to sell a new variety of basmati, Texmati, in the US and abroad. Actually, this new variety is derived by crossing Indian basmati with semi-dwarf variety and claimed as an invention or a new variety. Thus, it is a case of biopiracy and unfair biopatenting. Patents on turmeric, margosa, and many other Indian traditionally known as medicinal plants have also been attempted and awarded. Now we are fighting to cancel these illegal biopatents. We have won the legal battle against the patents of basmati and turmeric. There is a need for launching a genetic literacy movement in schools and colleges on the rapid developments taking place in the area of molecular genetics. It would help in better understanding of the opportunities and risks associated with recombinant DNA technology, right? This will help to promote the safe and responsible use of the tools associated with the new genetics in the country in the fields of food and agriculture, medicine, industry, and environment.